Good morning. For my journal club, I'll be talking about this article, Disruption of DNA methylation mediated cranial neural crest proliferation and differentiation causes orofacial cleft in mice. So this study was done in the School of Veterinary Medicine, Wisconsin-Madison University, USA, and was published in the Proceeding of National Authority of Science (PNAS) journal with the impact factor of 8.9. So following terminologies that we should know, cranial crest cells, they are multipotent embryonic cells that migrate to facial prominences and form much of the craniofacial mesenchyme. The DNA methylation here, it is a covalent addition of methyl groups to cytosine base and an epigenetic marker measured and manipulated in this study. The DNMT 1, that is DNA methyltransferase 1, it maintenance DNA methyltransferase enzyme was examined, genetically deleted in this study, and to test its requirement in palatogenesis. The DNMT3A and DNMT3B, they are de novo methyltransferase. The DNMT3B discussed in this study in the form of double knockout experiments. The maintenance versus de novo methylation. So DNMT1 and it is the maintenance. It preserves the methylation during replication, whereas DNMT3 it is performed de novo methylation. Conceptual background for molecular roles. The 5-azar-2- deoxycitrate, that AZAD-C, it is a pharmacologic DNMT inhibitor. It is a teratogen in this context, and it is used to reduce the methylation in vivo and in vitro. The palatal shelf and palatogenesis. It is the embryonic maxillary outgrowths that elongate and fuse to form the secondary palate. The primary morphologic process was analyzed in this study. The Clefts T system, a conditional knockout or CKO, it is a genetic tool, tissue and time specific gene deletion. In this study, they did for the SOX10-3 and OSR2 was used here. So what are they? They are the SOX10 derivatives for recombination and in early post-migratory craniofacial crest cells. The OSR2, it activates in later in the palatal mesenchyme used to dissect the timing of palatogenesis. The tomato reporter, it is a fluorescent reporter to visualize the recombination domains. The EDU incorporation assay, it is thymidine analog assay to label the S phase cells and quantify proliferation. The 5MC ELISA is ELISA based quantification of global DNA methylation used in this study on tissues and other cultured cells. The RNA sequence, it is a transcriptomic profiling used to detect differentially expressed gene in palatal shells after DNMT1 loss. So, false discovery rate, that is multiple testing correction applied to RNA sequencing. The Q was greater than 0.05 threshold was used. Gene oncology analysis, it is enrichment analysis to interrupt the cells of differentially expressed genes. The osteogenic or chondrogenic markers, so they use these markers to assess the differentiation defects. The O9-1 cell line, it immortalized cranial neural crest cell lines used for in vitro recapitulation and screens. The alizarin red and Elysian blue staining, so they used these two stain. the whole mount stain to visualize the bone using the alizarin red and the cartilage which was stained in blue by Elysian defects. The epigenomic editing, they used CRISPR tools to mentioned as a future approach to test the locus specific methylation effects. So coming to the novelty of this study, so this is the first study to provide direct in vivo and in vitro evidence that DNA methylation regulates the cranial Cleft cells proliferation and differentiation during orofacial development. It introduces DNMT1 conditional knockout mice and pharmacologic DNMT inhibition as models to study the cleft pathogenesis. They also established cultured cranial Cleft cells as a traceable model system to study environmental and epigenetic modulators of orofacial cleft. They try to bridge the gap between the epigenetic regulation and cellular mechanism that underlines the craniofacial morphologies. So the authors hypothesize that DNA methylation maintained by DNMT1 is essential for the normal CNCC proliferation and differentiation, and its disruption leads to orofacial cleft through the altered development of developmental gene expression and cellular behavior. So their aim was to determine how this DNA methylation regulates cranial Cleft cells biology and how its disruption contributes to orofacial cleft pathogenesis. So the objective, they genetically deleted DNMT1 in CNCCs using these recombinant mouse models to assess morphological molecular consequences. They also pharmacologically inhibited DNMT activity to mimic DNA hypomethylation in vivo and in vitro. They then characterized the effects of CNCC proliferation and differentiation using RNA sequencing, histology and marker expression. They then defined the critical developmental window during which DNA methylation disruption will cause the orofacial malformation. And finally, they established a CNCC model to study DNA methylation dependent mechanism and test environmental modulators, for example, Palate and cadmium. So in the methodology, they used the mice train genetically engineered mouse lines to achieve the tissue and time specific DNMT deletion. The animal husbandry, they mentioned that they maintain mice under specific pathogen free temperature and diet control conditions to ensure reproducibility. The animal study design, they perform time meetings and accurately determine gestational days for intervention and embryo fetus collection. The separation of mesenchyme and surface ectoderm, they used enzymatic separation to isolate epithelial and mesenchymal tissues from the maxillary process. Then they administered teratogen to pregnant mice at specific gestational days to inhibit DNMT activity pharmacologically, after which they measured the snout width and palatal shelf dimensions in fetus to quantify the craniofacial malformations. Also, they visualized DNMT1 and epithelial marker CDH1 expression by using immunohistochemistry to confirm the protein localization and pre-mediated knockout efficiency. Then they used the TD tomato in fluorescence reporter mice to verify spatial recombinase activity in the palatal mesenchyme, where it actually occurred in the palatal region. Then they quantified proliferating cells in embryonic maxillary mesenchyme by EDU incorporation to assess the growth effects of DNMT1 loss. Then they detected expression of osteogenic and chondrogenic markers to evaluate differentiation defects in the mutant embryos. The HME staining they did for general examination of general craniofacial morphology, palate and palate structure in late stages fetus. The bone and cartilage staining, they use the specialized stain which I have mentioned above to stain bone in cartilage. The bone was visualized in red color, whereas the cartilage in the blue color. And abnormalities, to visualize the abnormalities in DNMT1 deficient or AZADC exposed mice. Tissue microdissection and RNA isolation. They microdissected palatal shells from embryo for RNA extraction and transcriptomic analysis. The RNA library, they constructed RNA sequence libraries to identify gene expression changes following DNMT1 deletion. The RNAs they processed and aligned the sequence reads applied to DES sequence 2 and FBR corrections to identify differentially expressed genes. After which they quantified overall 5-methyl cytosine levels by ELISA to confirm DNA hypomethylation in mutant tissues and treated cells. Then they performed gene expression analysis where they performed quantified qPCR, quantitative PCR to validate expression of DNMT genes and lineage specific markers. The cell culture, they cultured the O91 murine cranial neural crest cells to model methylation dependent cellular processes in vitro. Lastly, they checked for the cranial neural crest cells osteogenic and chondrogenic differentiation by inducing lineage differentiation to test how DNMT inhibition alters the bone and cartilage gene expression. The statistical analysis they did here, unpaired t-test, paired t-test, one-way ANOVA plus Dermet, and false discovery rate. They did this to check for RNA sequence differential expression to correct for multiple testing across many genes. They did the mean plus SEM, and the data availability was mentioned they deposited RNA sequence data sets in the gene expression omnibus for open access. The results, the DNMT1 expression during palate formation and morphogenesis. So they found out that DNMT1 was detected in both epithelial and mesenchymal layers throughout the palatal development, suggesting its widespread regulatory role. The requirement of DNMT1 in cranial neural crest cell mesenchyme. So here we can see the image of control versus the conditional knockout of DNMT1, which shows there is a development of palate and it causes cleft palate and other craniofacial malformations and defective palate fusion. The temporal restrictions of DNMT activity, so disruption of DNMT1 function at early developmental stages around 9.75 produced orofacial clefts, while later inactivation did not produce, indicating there is a critical period for DNA demethylation. Reduction of DNA methylation in DNMT1 conditional knockout embryos. So global DNA methylation levels were significantly reduced in maxillary mesenchyme of DNMT1 deficient embryos during the sensitive and developmental window. The impact of DNA methylation on CNCC proliferation and differentiation. So DNMT1 loss led to reduced proliferation and impaired osteogenic and chondrogenic